we are preparing for the SeekOneTM single-cell multiome ATAC plus RNA experiment. Now that droplet PCR is complete, we'll immediately proceed to emulsion break. Thaw the reducing buffer on ice, vortex, and spin briefly. Freshly prepare 80% ethanol. Bring cleanup beads and size selection beads to room temperature. At room temperature, add 3 microliters of buffer S to each tube containing droplets. Then add 100 microliters of demulsion agent and incubate at room temperature for 2 minutes. After demulsification, 2 phases are visible. The upper clear aqueous phase contains the target molecules and must be recovered. The clear bottom phase is oil and should be discarded. Slowly aspirate about 130 microliters from the bottom of the tube. Leave 2 to 5 microliters of the oil phase to avoid sample loss. If the aqueous phase still looks cloudy, demulsification can be repeated. If it remains cloudy after a second round, proceed directly to cleanup. After demulsification, the product can be stored at -80 degrees for up to 1 week. Vortex the cleanup beads for 30 seconds. Spin briefly. Prepare the cleanup mix according to the manual. Add 186 microliters of cleanup mix. Gently pipette at least 15 times, letting the tip move with the liquid. Incubate at room temperature for 10 minutes. Place the tube on the magnetic rack until the solution clears, then remove the supernatant. Wash the beads twice with 80% ethanol. After removing residual ethanol, air dry at room temperature. Beads should look matte, not cracked. Over-dried beads can reduce recovery and affect cDNA quality. Elute the DNA with nuclease-free water. Incubate for 5 minutes. Place on the magnetic rack until the solution clears. Then transfer the clear supernatant to a new PCR tube. Emulsion breaking and cleanup are complete. The purified product is now ready for pre-amplification and library preparation. In the following steps, the purified ATAC and cDNA fragments are amplified and prepared for library construction. These steps follow standard library construction procedures and are shown here at a conceptual level. In Step 4, ATAC fragments and cDNA are pre-amplified together to generate sufficient material for downstream processing. After pre-amplification, the product is split into 2 parts, one for ATAC library construction and the other for gene expression library preparation. For detailed volumes and incubation conditions, always follow the user manual.